Finding an ancestor usually meant digging for bones or stone tools. But the physical fossil record eventually hits a wall, ground into dust by the earth. What emerges from two recent articles is a different way of looking backward. In The Atlantic, Ross Andersen shows how researchers use DNA sequencing and laser mapping to locate missing human ancestors and lost ancient cities. A Smithsonian Magazine piece goes back much further, reverse engineering the enzymes inside modern microbes to find the chemical sparks of early cellular life. Read side by side, a surprising shift becomes clear. Instead of relying on what the dirt left behind, scientists are reading the genetic code of living things to reconstruct a past the planet erased. An article published just last week in Smithsonian Magazine traces the chemical origins of life by reverse engineering the enzymes inside modern microbes. Listen for the description of Earth's earliest common ancestor. Before cells could make their own energy, this ancient organism had to rely on loose metals in the ocean water to run its basic reactions, capturing the exact moment when raw geology started turning into biology.
A new study points to two origins of life on Earth by tracing early chemical reactions in single-celled organisms. The work challenges the assumption that all life descended from one free-living cell. Instead, it suggests that two microbial lineages, the bacteria and archaea, evolved independently from a primordial, non-living ancestor. Among biologists, few questions have been debated as fiercely as the origin of life. How did self-sustaining organisms, capable of transforming molecules to capture energy, arise on the primitive Earth? Before any cells existed, scientists suspect chemical reactions occurred around deep-sea hydrothermal vents, where heat and high pressures led to spontaneous chemistry that was sped up or catalyzed by metals in Earth's crust. One possibility for the origin of life is that, over time, these relatively simple geochemical reactions evolved into the reactions that create the building blocks of life. Living creatures rely on a core network of about 400 metabolic reactions that are catalyzed by proteins known as enzymes. This core metabolism creates energy and makes key materials for cells, like nucleic acids, amino acids, and vitamins. Now, a team of researchers has retraced the steps for how this essential metabolic network was assembled in a paper published today in Science Advances. One of their most striking findings, life on Earth likely evolved not once, but twice. This just opens up a whole lot of questions in terms of what is alive, what is life, what is possible, and what we might find elsewhere, says senior author William Martin, an evolutionary biologist at the University of Dusseldorf in Germany. All organisms alive today descend from the last universal common ancestor, or LUCA, that likely dwelled in the early oceans. This mysterious ancestor, the new study suggests, was still reliant on its environment to conduct metabolism. Rather than fueling its energy-producing reactions on its own with enzymes, LUCA depended on small organic molecules and metals in the environment to conduct about half of the core metabolic reactions, the researchers found. Then, over time, some of these metals were replaced with more specific and efficient enzymes as the proteins evolved to do the same jobs. What's so cool about this is we're probing a phase of evolution where new enzyme activities are still arising, replacing geochemical reactions, and giving rise to coalescence of metabolism in the ancestors of archaea and bacteria, Martin says. Archaea and bacteria are two of the three domains of life, and these microbes arose early in Earth's history. The third domain, eukaryotes, the one that animals, plants, and all organisms with nucleus-containing cells belong to, evolved later from a hybrid of the first two groups. For many years, scientists contemplated three possible scenarios for the origin of life. Either bacteria evolved from archaea evolved from bacteria, or both independently evolved from LUCA. The new study makes the argument that the last possibility is the most likely one. Quick fact, archaea and bacteria. While bacteria live in diverse environments, archaea thrive under extreme conditions, like high heat and acidity. Scientists can illuminate how bacteria and archaea emerged by comparing when their ancestors evolved certain traits
and pinning down which of those traits might have been inherited from Luca. Earlier work did this with ribosomes, the parts of a cell that produce proteins. It showed that Luca had primitive ribosomes that were composed of 33 proteins.
Then, from Luca, the bacterial lineage evolved 21 unique ribosomal proteins, while the archaeal lineage evolved 29 unique ribosomal proteins. Each lineage, in essence, independently continued tweaking and enhancing how its ribosomes worked. For their latest investigation, focused on reconstructing metabolism, Martin's team took a similar approach. This time, they examined the genes for metabolic enzymes in modern bacteria and archaea to reconstruct their evolutionary history. Just as some elements of the ribosome were universal across the two groups, while each lineage also evolved its own ribosomal proteins, we realized, looking at our metabolic genes, that this is a similar pattern, says lead author Natalia Mrnjovac, a graduate student in Martin's lab. They first worked to narrow down which enzymes Luca was using and which of its metabolic reactions required metals from the hydrothermal vents. The team reasoned that each reaction in this primitive metabolism either used elements available in the environment or elements generated by other reactions in the system. Over time, these reactions could build on the previous ones, leading to new biochemical possibilities and an increasingly complex metabolic network. The key was to identify which metabolic reactions had to come first and which subsequent ones depended on which others. It sounds like it would be hard, with hundreds of reactions, to work out the sequence in which they developed, says Mike Steele, a mathematician at the University of Canterbury in New Zealand and a co-author of the study. But it turns out there's a fast algorithm for doing this. The team then found that bacteria and archaea shared some enzymes with Luca, but each lineage also had some that were not present in the ancestor.
Essentially, bacteria and archaea independently evolved different enzymes to replace the same metal-catalyzed reactions. This shows the reaction is older than the enzymes that catalyze it, says University of Ottawa chemist Joseph Moran, a co-author of the study. These distinct solutions to the same metabolic problems are evidence to Martin that bacteria could not have been derived from archaea, or the reverse. The simplest interpretation, he says, is that there were two independent transitions from Luca to free-living cells. Not all researchers agree on this description of Luca. Some have proposed that Luca was a more complex cell with simple membranes that were perhaps made of a mix of archaeal and bacterial fats, or lipids. Then, this blended ancestor would have eventually diverged into the archaeal and bacterial lineages in only one origin of free-living cellular life, explains protein crystallographer and origin of life researcher Juan Fontechilla Camps of the Institute of Structural Biology in France, who was not involved with the new study. But Martin argues that this complexity is unlikely. Bacteria and archaea use distinct methods for making the lipids in their cell membranes, he says, so they probably didn't both inherit those mechanisms from Luca. They really are totally different, adds Sonja Verena Albers, who studies the biology of archaea at the University of Freiburg in Germany and was not involved in this work. You need a totally different setup of genes or enzymes to make the different lipids. Based on its metabolism, too, Martin argues that Luca was a simpler ancestor, something not completely alive.
There can be no question that free-living cells are alive, says Martin, referring to cells that are capable of growing, dividing, and surviving on their own. We can all agree on that. But Luca was still reliant on small organic molecules and metals in the Earth's crust to carry out metabolism. When it comes to being classified as truly alive, that's not going to cut the mustard, he says. Martin has long argued that metals were the original catalysts for metabolic reactions, and that over time, those metals were replaced by enzymes. There's strong support for the idea that geochemistry would have led to life's biochemistry, says Donato Giovanelli, a microbiologist at the University of Naples Federico II in Italy, who studies the origin of life. For instance, many enzymes have metals in their catalytic sites, where the chemical reactions occur. And in the last several years, Martin, Moran, biochemist Martina Preiner, and their colleagues have shown experimentally that many of the reactions used in core metabolism today can indeed proceed when they swap enzymes out for metals, suggesting that they could have been catalyzed by metals before those enzymes evolved. One of the most important metabolic reactions creates the energy-carrying molecule of cells,
essentially a tiny battery that powers them. Today, this molecule is primarily ATP, but the primitive oceans did not have ATP for Luca to use. Martin's team revealed how, in this ancestor's primordial metabolism, this key reaction could have been catalyzed by metals. They found that phosphite, a form of phosphorus, could react with organic molecules with the help of palladium, a metal in the platinum group of metals. This reaction allowed Luca to store energy as ADP, a precursor to ATP.
The ability to transiently store energy was game-changing because it could power many other metabolic reactions that need an input of energy, such as the formation of peptide bonds between amino acids, or the activity of ribosomes during translation of RNAs into proteins. This reaction paved the way for the shift from geochemical to metabolic reactions and explains how phosphorus became such a central player in our own biochemistry. That's one of the key discoveries in this paper, says Giovanelli, who was not involved in the new study. Reconstructing early metabolic reactions, as the team did in the new paper, is a valid thing to do, Fontecilla-Camp says. The question is whether that's the way it really happened or not, and that's very difficult to know. Giovanelli says that the team has produced a strong paper that puts forth a different line of thought and brings together things that had not been connected before. But he suggests thinking about their conclusion as a possible scenario for the origin of life, rather than a definitive answer. We can reconstruct how plausible things might be, but it's going to be almost impossible to know exactly how things happened.
The Smithsonian piece leaves you staring at a deep-sea vent, trying to picture an ancestor that wasn't quite alive. The writer upends the classic textbook image of a single, independent cell sparking life on Earth. Instead, you get a messy, incomplete chemical reaction that had to happen twice. But the really striking detail is how researchers figured this out. With no 4 billion-year-old fossils to dig up, they had to read the metabolic code of microbes alive today. Ross Andersen, up next, picks up that exact same tool, applying it to a much more recent family tree. His piece jumps forward billions of years from those oceanic vents to Ice Age Europe, tracing the origins of modern humans. Just like the biologists studying early cells, the researchers Andersen follows are bypassing the physical fossil record. When the dirt erases the bones of ancient hominins, the genetic code of living people becomes the only archive left. Read alongside the first piece, Andersen's reporting on ghost ancestors, ancient people who interbred with humans but left no physical trace, feels less like a standard archaeology story and more like a massive forensic accounting trick. Both pieces show how the past never really vanished. It just hid itself inside the bodies of the living.
The human origin story is being radically revised. New secrets from our past have been coming out in droves. Some 50,000 years ago, modern humans wandered into Ice Age Europe. The continent was new to us Homo sapiens, but not for beings of our general kind. Neanderthals and their ancestors had been living among Eurasia's glaciers for ages, and in ways that would have struck us as familiar. Evidence suggests that they may have strung eagle talons together into necklaces and stained their bodily ornaments with pigments. They tended fires, cooked food, and maybe even made their own glue. On sandstone cave walls, they etched wavy lines, dots, and other geometric designs. After first contact, under social circumstances that we cannot possibly imagine, modern humans and Neanderthals had children together. Around the same time, in Asia, our species also procreated with another species, the Denisovans. Scientists know this because they have sequenced DNA from Neanderthal and Denisovan remains, and they can see fragments of their code in our genomes to this day. Now we are learning that still more of these fusions of blood and culture may have shaped our lineage. Last week, a team of researchers from UC Berkeley and Johns Hopkins University published new evidence in Science, suggesting that 2 ghost ancestors haunt our family tree. Not Neanderthals or Denisovans, but some other hominin populations. DNA analysis is only one of the techniques that scientists have been using to cast narrow shafts of light into the dark abyss that is the human past.
Researchers from several disciplines have been collecting and reinterpreting ever smaller traces of our forebears to reimagine how they lived, what they made, and what family secrets they might still be hiding.
The Ghost Ancestors paper is the most recent example. The team from UC Berkeley and Hopkins analyzed some very odd sequences of DNA in the human genome, which were discovered 20 years ago. These don't match up with those of any ancient hominin species that have already been sequenced, and their origins have been mysterious. But now we have some suspects. One of the strange sequences appears to come from a hominin who mated with us before we ventured out of Africa. The other may come from the great wanderer Homo erectus, who left Africa nearly 2 million years ago and ranged across Asia. Homo erectus may never have encountered Homo sapiens directly, but the researchers suspect that the hominin interbred with the Denisovans before the Denisovans interbred with us. Either way, all of these people, if it is right to call them people, played a role in our species' story of becoming, yet we are only now learning the nature of that connection. All year, scientists from different fields have been making revisions to the human origin story. In January, one team announced that they'd found handheld wooden tools along an ancient lakeshore in present-day Greece, a refuge between glaciers where hominins of some kind butchered elephants about 430,000 years ago. These are surely not the first wooden tools ever to be made, but they are the most ancient that we've managed to find, and they were used during the Stone Age. That name, Stone Age, is misleading. It reveals more about the limits of archaeology than about the material culture of the people who lived through the period, who may not have thought of themselves as being defined by stone tools at all. We perceive them that way only because their knapped axes of flint and obsidian are likelier to have survived to the present day than the digging sticks that our forebears smoothed and sharpened. Beyond stone and wood, these lakeside elephant butchers likely put all kinds of natural materials to clever use. They may have employed woven twine to sew animal hides together, but we wouldn't know it because twine unravels into thin, disintegrating filaments, and bacteria rapidly remove hides from the world. Wood tends to rot away too. In this location, the tools had the good geological fortune to be buried quickly under mud. Stone tools have themselves been reinterpreted. Researchers recently detected traces of poison on an ancient set of quartz arrowheads that were dug up in 1985. The weapons were found in a South African rock shelter that was used by modern humans 60,000 years ago, tens of thousands of years earlier than what had been regarded as the oldest confirmed human use of poison. The ancient South African archers retrieved the neurotoxin from a plant bulb that grows as large as a honeydew melon and dabbed it onto the cold stone of their arrowheads, knowing that it would slow down any animal that they pierced. Some scientists are now calling for a reappraisal of other ancient arrowheads to see whether they too had been sheathed in poison by people whose ingenuity had previously been underestimated by science. Piecing together the history of our species in this way is tedious work because the evidence is so fragmentary. The Earth's violent, self-renewing processes erase so much. During the planet's cold spells, its glaciers steamroll from the poles toward the equator, grinding and scraping whole land masses clean. In its warmer, middle zones, jungles can sprout up within decades, covering human ruins in a thick and devouring layer of green. Scientists have recently learned that the Amazon rainforest has been concealing millennia-old earthworks on an enormous scale. A team of international researchers spent weeks flying LiDAR-equipped planes back and forth over a swath of Amazonia. Their laser scans revealed nearly 400 immense structures that were previously unknown to science. From the sky, they saw that these systems of raised banks and deep ditches formed geometrically precise circles, octagons, and squares, and were linked to one another by remarkably straight roads. The earthworks were built 1,200 to 2,500 years ago by the Akwari civilization. Having surveyed only one small portion of Akwari territory, the researchers suspect that tens of thousands more of these structures may be waiting silently under the region's trees. 100,000 years from now, the Akwari ruins might vanish altogether, and in Earth time, 100,000 years is nothing. In 10 million years,
Even the skyscrapers and continent-sprawling infrastructure of our own civilization will be crushed into sedimentary layers. Nearly everything that we have created will be reduced to trace elements and isotopes, but for a few specimens lucky enough to be fossilized. For all of the other details of our world to persist in human memory, science would need to get better, or civilization itself would need to keep existing and telling its own story from now until then.
When the dirt yields no bones and ancient ruins wear down to dust, the search for the past usually stops. What lingers is the realization that while the Earth easily crushes its physical history, the instructions survive. The deep past is still walking around, hidden inside the cells of modern people and the microbes in the soil. It leaves a quiet question to sit with. If the blueprints of erased ancestors are written into the living code of today, what other vanished worlds are breathing right now in plain sight?